So this is what it has to be explained in the video part. What is packaging of DNA? So I'll explain this thing first. So the DNA is negatively charged and histones are positively charged. So this negatively charged DNA binds itself in around these histone molecules, and then in turn these binded histone molecules which are surrounded by DNA coils around itself in a structure like this, and then it forms a structure called as chromosome. This is what is to be explained or make uh, children visualize this. So it's all about this. The purpose of this video will be these two mainly points: why does the DNA has to package and how does it happen. So why? Because DNA is like its length of a DNA is 1.2 meters and the nucleus radius is 10 is to minus 6 meters, which is like a very small structure. So hence the DNA has to coil and package itself. It becomes necessary because it has to fit themselves in the limited space of the nucleus. So this answers the question. So we'll have to show the DNA is this much of diameter, there are 23 base pairs, so nearly 20 trillion meters has to fit inside this small nucleus. So the only thing that can happen is coiling around itself, making a complex structure called as chromosomes, which can fit inside the 10 raised to minus 6 meters nucleus. So sorry. So this answers the question of why. And how does that happen? How happen is there are positively charged histone protein due to presence of positively charged amino acids. We can show some structures and name it amino acids there. So uh, as histones are positively charged, DNA is negatively charged. They wound around it. Two each of four core histones exchange make up to form a octamer. If we have seen here, so it's an octamer. 8 molecules of this type of histones are formed, and then the DNA coils around itself. Like these are the four core histones. So we'll have to name them. Like let's show some callouts wherein we can mention that these are the four types of histones. And the naked DNA gets founded around nucleosomes. H1 is connected at the place where DNA enters and leaves these nucleosomes. So we can show that. So DNA wounds around nucleosome 1 and 3-4 times the later gets around next nucleosome. So just a minute. So this is the one spiral, and the second is like just the 3/4 of the histone octamer which has formed. DNA wounds around nucleosome 1 and 3/4 times the later gets found to next nucleosomes. The DNA which wound around nucleosome is called core DNA and is of 46 base pairs. BP is base pairs. And the DNA which links one nucleosome to the next nucleosome is called linker DNA. We'll have to show. So we can maybe differentiate using these colors. H3 is of blue, H2B is of green, and we can show like this. After the wounding of DNA around nucleosomes, it looks like bead on a string structure which is of 10 nanometer in diameter, like this. Now the structure starts coiling around another type of protein called non-histone proteins. So this is an histone protein, these are non-histone proteins. So this starts coiling around these non-histone proteins. Now the supercoiling give rise to a solenoid fiber, like solenoid looking fiber of 30 nanometer diameter. So then the solenoid, it all coiling around itself. Now the solenoid also coil around itself in a way, like structures called these mini bands. So these mini bands get stacked on each other like stacked mini bands, and now these stacked mini bands look like a chromosome. So we'll have to start from a DNA. I'm so sorry. Start from a DNA helix, double helix structure. We'll have to show that, which coils around in histone proteins and which are positively charged, and then these coilings happens. Now these coiled also get coiled in a way that it gets picked into this chromosomes. So this is what has to be shown. I'll share few of the video references. uh, Maybe that can help you even more. Yes.